Sono a Isaccia e dietro di noi vediamo l'Ucraina e questo
Can you hear my voice now? Wait, hold on. By the way, you have a problem with the camera. You hear?
Okay.
There's a
Just
delay.
the delay.
Yeah, exactly.
Now it makes sense.
Feedback form. Join the Blue Spirit and become part of EMS Torino. Health. Europe. Together.
Test, 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 test.
Only at the end, I would like to let you see a video on YouTube.
Yes, of
Is
course.
that a problem?
No, no, we will manage it when we get to it.
Okay.
Okay.
So I just call you.
Yeah, we will be here.
Okay. Is there anyone in presence, or it's only? Only the three of you. Okay, just. No problem. If there are three students, I will make a presentation for three students. It's not a problem on my side. It will be only for a few implements.
Would it be okay if later on we make the presentation available online always?
Online on which platform?
On the server?
Yeah, sure. Yes. On YouTube, you mean? Yeah. Okay. When you wish to start?
Okay, so good morning, everyone, and thanks for the people present in the class for attending, and whoever is with us online, also welcome. I hope you're having a wonderful morning up till now. As a small introduction, not to take a lot of time from Professor Chivra, this is our second seminar for the year. It will be on entrepreneurship in medicine. Professor Chivra will go into details of all the steps needed to get a drug or any medical product from zero to the market. And I believe we all know Professor Chivra. We've been taught by him microbiology, and he's a microbiologist, and he has been working in, I think, several startups, including an antibiotic with an innovative idea. So not to take much more time, Professor Chivra, we can go on.
Welcome to everyone. Can you hear me? Doesn't. Okay. Okay, again, welcome you to this speech. I will talk to you about which are the main aspects of drug development and what is needed to, which are the steps to set up a business or a startup in the pharma sector. Just to introduce you, I'm basically a researcher. Okay, so I'm an academic. So basically, my main topic of research is antivirals. This is my, good morning. You're welcome. This is my group. I work since it's something like 17 years with Professor Lembo and Professor Donalisio. At a certain point, and this is the reason why we are here today, at a certain point of my career, I started a research about a specific family of molecules, which are called oxysterols. Oxysterols are physiologic molecules. Some of them are spontaneously produced in our body, starting from the cholesterol. They are involved in a wide spectrum of cellular and physiological processes, just like, for example, cholesterol homeostasis, bile acid synthesis, and so on. And they were actually considered at the beginning of the 21st century only as byproducts of the metabolism of cholesterol. But we discovered that oxysterols are broad-spectrum antivirals. Usually antivirals have a very specific target, are virus-specific. So the dogma is one antiviral for one virus. There are no broad-spectrum antivirals in commerce. So when we discovered for the first time this specific feature of oxysterols, we imagined that we had an opportunity to develop a novel class of molecules from the drug discovery to the drug development. The first thing you have to know is that to do it, which will be the main topic of this speech. The first thing to remember about this is that if you want to develop a business, and specifically, if you want to develop a pharma startup, a small company, who has the aim to develop a novel drug, you must step out from your comfort zone. This is a totally different job from the one you are planning to do. You will not be physicians anymore, or in my case, you won't be a researcher anymore. We'll start a completely new job. You must learn a completely different dictionary, novel notions. You must get visibility into a totally new community, which is made of investors and industrial partners, which is totally different from the network you have for your specific activity. You must get able to fundraise, so to search funds to fulfill the financial needs of your startup and so on. But again, let's see which are the steps from the very beginning to develop an idea. The early beginning is always the drug discovery. In our specific case, we were studying a panel of natural molecules, okay? But the very beginning, in some cases, can start from the identification of a specific target, of a specific protein, which is involved in the pathogenic mechanism of a specific pathology. The very beginning sometimes starts from, in other cases, starts from a panel of synthetic molecules, and the active molecules are identified by serendipity. So the very beginning has many, many different starting points. But this is always what is in the beginning of our scientific process, the hypothesis. Does your molecule work? Is it active against, in our specific case, against a specific viral pathogen or not? So you make a research, so you make experiments, you gather data, and then you draw your conclusions. Then usually what happens in the academic environment is that you disseminate. So you share your results with the community. There are several channels, congresses, both by oral communications or posters, or you publish your research on peer-reviewed journals. But what happens if you have an opportunity? So if you want to exploit an opportunity to develop a company and develop a business, so to transform your scientific discovery into something that is valuable, in this case, you must not disseminate. You must be silent. You must not disclose your data in any way, because you may want to patent it. So usually when you discover a molecule which is endowed with a particular pharmacological activity, the first thing to do is to be silent and patent. So submit a patent application. What is a patent? It's basically a legal protection. It's a sort of barrier. It's a sort of entrance barrier. When you submit a patent application, no one else will make, use, or sell your specific product, even if it's not yet a product. No one will use, sell, or produce your same molecule. In order to be patented, a scientific discovery in general must have three different features. It must be novel, so previously it must have not been disclosed. It must be useful, so it must be used as an industrial level. It must solve a problem, must have an impact on a specific pathology in a case of a drug. And it must be not obvious. This is the most critical point, and it's the less intuitive point of a patent. Your discovery, in order to be patented, must not be obvious to a person having ordinary skill in your same discipline. For example, in our specific case, it was known that oxysterols were active against a specific class of viruses, the enveloped ones, the ones that are endowed with a pericapsid. We patented the use of oxysterols against the other group of viruses, the non-enveloped ones. So the question in this case, was that obvious that the oxysterols could work also against another category of pathogens? Is it clear? So the point here is that the molecule, the activity of the molecule, the molecule that you want to patent maybe is already demonstrated as active against other pathologies, okay, or against other pathogens in our cases. But maybe it's still patentable. So a person with ordinary skills in your same scientific discipline may not find obvious that your molecule can work in the pathology you want to patent. Another point here, it's useful, which means that you cannot patent a natural phenomenon. So for example, you cannot patent, I don't know, an intracellular signaling pathway. Your patent must be useful. So this is the correct path to follow if you want to patent your discovery. You discover something, the activity of a molecule or a novel molecule, then you patent, and then you disseminate. If you disseminate, then you cannot patent. This is obvious. It's not novel anymore, your discovery. You can patent, in terms of drug development, you can patent a novel chemical entity, so a totally novel molecule. In this case, you are patenting the structure itself of the molecule, which was not possible with oxysterols. They are natural molecules. Their structure is already known. You can patent an improvement to an old product. So for example, you can patent a novel formulation that improves the pharmacological activity of your active ingredient. So you can patent a combination of excipients that increases remarkably and significantly the pharmacological activity of a known active principle. Or you can patent a novel use for an existing or old product, as in the case of oxysterols. This is widely used as a patent. It's called, and it's widely used as a strategy. It's called repurposing, which was a strategy of drug development which was highly exploited during the pandemic. Old active principles used for a novel pathology, which is the COVID-19. So why you have to patent in the case of drug development? The answer is that in most cases, during a drug development process, the patent is your most important asset. So the value of your company, most of the times, relies only on the possibility that other people cannot use your same molecule, or cannot use your molecule in the same way you are planning to use it. So for the same pathology, for example. So, and if you have any questions, just ask. Yeah. Oh, the microphone.
So at what point are you supposed to patent? So when you first discover the molecule, or you have to wait until you know its full capabilities, or until you finish your whole spectrum of research on it?
You must patent at the beginning of your development process. So basically, if you are a chemist and if you have synthesized a novel library of unprecedented and not before disclosed molecules, you have to patent them if you plan to exploit them for industrial purpose. Or if you discover a novel use of an existing molecule, again, you have to patent this discovery at the very beginning. The reason why is that if you do not patent, you usually cannot disseminate your discovery, which blocks your academic career most of the times. But you have to patent only if you foresee a strategy to develop your patent. So only if you foresee practical use of your invention. If not, it's totally useless to patent, even if your discovery is patentable. The reason why is that a patent is expensive. So if you do not plan to exploit your patent at an industrial level, you only have a cost in your hands and you have to pay for it each year. So it's totally useless. Yeah.
In the case of using old drugs to develop, like to find new ingredients to make them more potent, they have obviously been patented before. How can we proceed knowing that? Do we have to like buy rights of the old, like exploit the patent of the old drug so that we can work on it and then repatent it with the new ingredient?
Usually at the beginning you have no money to pay your patent, so you patent only if you have something exploitable, so useful, novel, and not obvious. If your discovery is already covered by another patent, just stop. Don't patent, don't do anything else, write your research paper and disseminate. And move on. Maybe you will discover a novel structure of your molecule that it's more active, a novel formulation that is more active. So you can, my advice is to move on with your research in that case. Patents are expensive. This is the point. So at the beginning of your process, you are just a researcher. And your funds are useful to fund your personal research, not to pay the patents. So if someone else has already done something on your same topic, has already patented, don't do anything, don't patent. You usually don't want to buy a patent. It's something that it's totally useless at that point. So it's a source of recognition for the investors. So usually the patent is the only asset of your company, at least in the drug development area of startups. Also, the synthesis process of your molecules can be patented if it's not, if it's novel, if it has not been disseminated before. The patent provides you with a protection against imitators, and then it's an incentive to develop a commercial product that can be, in turn, sorry, I have a problem. That can be sold or licensed to other companies. A license is a sort of rent of your patent. So, how to move from the drug discovery to the drug development, and how this process is associated with the business of a pharma startup? The answer to the second question is that the more you develop your active principle, so from the active principle to the drug, the more you develop your product, the more the patent acquire value, okay? It's a sort of de-risking project, de-risking strategy. So this path is particularly fundamental to understand. The more you fulfill the milestones on this development process, the more your patent acquire a specific value. So let's say something about all the steps. The first step of drug development is always the discovery, as we said. And talking about specifically the antivirals, usually you start with a panel of molecules that you want to test. This was the our first publication about oxysterols. In our specific case, this is just an example to let you know. We tested 5 different molecules. There are libraries that are made of tens or thousands of molecules. You test your molecules, and at the beginning of your process, so the step 1 of drug discovery is to obtain, to calculate, usually in vitro, the EC50, so the effective concentration that provides the 50% of your effect. In the specific case of antivirals, the concentration that inhibits the 50% of infectivity, usually. The second parameter that you want to understand in this first phase is the CC50, cytotoxic concentration 50. So the concentration of your molecule that in vitro kills the 50% of the treated cells. In general, the lower is the EC50, the better it is. The higher it is the CC50, the better it is. Both these values in this step of drug development are fundamental to calculate the SI, which is the selectivity index, which is basically a ratio between the CC50 and the EC50. This is the first step. So a sort of proof of principle, mainly in vitro, of your molecule. The second step is to assess, during the drug discovery step, is to assess the mechanism of action of your molecule. So how does it work? There are basically a couple of things to understand, talking about the mechanism of action. To identify the molecular target of your molecule, so which is the protein that it's targeted, a protein in general, that it's targeted by your molecule that triggers, in this case, your antiviral activity. And the second one is to characterize the effect, for talking about antivirals, on the viral replicative cycle, which means usually to understand which is the step of viral replicative cycle that it's inhibited by your molecule. This is an example of what we have done with oxysterols. This is the mechanism of action. I'm going to describe you the mechanism of action of 2 oxysterols against the rotavirus, which is the most widespread cause of infective gastroenteritis in children. Usually these viruses enter the cell by endocytosis. So they are endocytosed, they reach the early and then late endosomes, and then they exit. We found out that our molecules could inhibit the exit of human rotavirus from the late endosomes. So the viral particles were sequestered inside the late endosomes, and these blocked the replication of the virus. But more in detail, the target we discovered, the target of our molecules was a cellular protein. Oxysterols are host-targeting antivirals. The protein was the OSBP, the oxysterol binding protein, which usually interacts with the second protein on the ER, which is VAPA. This interaction regulates the recycling of cholesterol, the transport of cholesterol from the membrane of the ER to the membrane of late endosomes. Basically, what happens when you treat the cells with oxysterols? They bind to OSBP, inhibit its interaction with VAPA, we have a block of cholesterol recycling and an accumulation of cholesterol on the membrane of late endosomes. This modification blocks the exit of human rotavirus into the cytosol from the late endosome. This is a characterization of a mechanism of action. This is an information you have to gather at the beginning of the drug development. Questions? Yeah.
We have to gather this information before we file for the patent or after?
Even after. But in general, these informations must be gathered at the very beginning of your project. Usually recognizing, identifying a specific molecular target allows you to make predictions on the toxicity of your molecule. The more specific is the interaction of your molecule with a molecular target, the less off-target effects you will have. So this information is better it is better to have this information at the very beginning. Not necessarily you have to obtain it before the patent. Sometimes it comes after. But it's useful to obtain it before. It's, this information has more to do with the scientific profile of your project than with industrial development. And then in this case, you may want to gather other informations of your molecules. For example, we studied the effect of oxysterols on the cellular proteome. We identified the presence of oxysterols in several biological fluids. We discovered that one of them, the 27-hydroxycholesterol, is particularly concentrated in human colostrum, more significantly more than other oxysterols, which suggested us to identify 27-hydroxycholesterol as our main molecule, our most promising molecule. We discovered that 27-hydroxycholesterol is active also against the SARS-CoV-2 virus, and then that severe COVID-19 patients, for some reason that we didn't have dissected so far, severe COVID-19 patients have a significant lower concentration of 27-hydroxycholesterol in their blood if compared to control patients, so to non-infected and non-symptomatic, obviously, patients. So in this step of your drug development process, you may want to gather the more information as possible about your molecule, which provide you information about the efficacy, but also about the safety of your molecule, that inside a panel of molecules can indicate which of them is the most promising one. So in this step of your development process, the main topic is the research, not the development, okay? Not the industrial development of your molecule. With step 2, everything is different. You start the preclinical research if you plan a possible use of your molecule as a drug. In this step, you may want to gather information about the safety, the efficacy, and the toxicology profile of your molecule. So preclinical research is about pharmacokinetics, pharmacodynamics, and toxicology. First, you want to characterize the ADME profile of your molecule. How is it absorbed? How it distributes in an organism, in a complex organism, how it is metabolized in order to be eliminated. Usually these kind of tests are performed in animal models. In vivo pharmacodynamics has two specific aims: to obtain the effective dose 50 and the effective dose 95. So the dosage of your molecule, which is the dose of your active ingredient that provides you with a 50% of effect in vivo. And which is the best way to administer your molecule. Is it an oral route or is it an injection route? These two steps provide you with these informations. These two informations are fundamental in order to plan a development of your molecule, are not dispensable. In vivo toxicology can provide you with several informations, but the most important one is the lethal dose 50. So the dose that, once administered, kills the 50% of treated animals in this case. Once you have gathered all these informations and the previous ones, so the ones from step 2 and step 1 and step 2, if you find that your molecule is a promising one, so if you plan to develop your molecule, you may want to start, you have to start the clinical trials. In order to start the clinical trials, you have to gather this information and write down an IND application, an investigational new drug application, which is a mandatory document which is requested by regulatory bodies, just like, for example, the FDA in the US or the EMA in Europe. This application, this document, must contain all these informations about the animal pharmacology and toxicology, so the preclinical data, which are mandatory, and must be obtained in a regimen of good laboratory practices, GLP. The manufacturing information, so which is your company, which is the company that produces your active principle, how do they produce it, so the synthesis process, which is the composition of your active ingredient, which is the manufacturer, which is the stability, for how long it is stable and at which temperature your drug is stable, and so on. And then you have to write already here in the IND application the clinical protocols you are planning to follow, together with information about the qualifications of clinical investigators that are going to manage these clinical trials. Usually is a physician that submits the IND application. Once you have prepared, once this document has been prepared, it can be submitted to FDA or EMA, and they make a review, they make an evaluation and assessment of your IND and provide you with, eventually, sometimes, they provide you with a permission to start the clinical trials. One thing to say at this point is that each one of these steps is not dispensable. Okay, this is always the path of a drug development. These are always the steps of a drug development process. With clinical trials, the most part of the problems begin. Okay. Usually, the most part of the pharma startups fail in these steps. more frequently during phase 2 clinical studies, but I will tell you why. Phase 1 clinical trials are the first studies in human. The purpose is to assess the safety and the dosage in humans of your molecule. So usually in these trials, healthy people are recruited, okay? Usually a small number, tenth of people, okay? This is the less expensive clinical trials between all of them, and I would say the less risky, okay? Because you already have the preclinical data to make some predictions about the safety of your molecule. Phase 2 clinical studies are the ones that want to assess the efficacy of your molecule in human beings. In this case, patients are recruited. This is a huge problem with infectious diseases. At least for some infectious diseases. For example, in our specific case, we wanted to develop a drug for rhinovirus infections, which is not a chronic infection. You do not always have patients available. The diagnosis of a specific rhinovirus infection usually is not done. So it's very difficult to gather these specific cohort of patients. This is just to tell you that it's very hard sometimes to design a phase 2 clinical trial for an antiviral drug specifically. Usually in this case, 100, 200 of patients must be recruited, but it depends. It depends on the epidemiology of your of the pathology that you want to target. Phase 3 clinical studies have the purpose, the aim to dissect the efficacy and to monitor the off-target, the adverse reactions of your molecules. The difference between phase 3, the main difference between phase 3 and phase 1 and phase 2 clinical trials are the numbers. In this case, thousands of people must be recruited to gather statistically significant data. Usually what happens with pharma startups, it's not a rule, okay? It's something that usually happens, that pharma startup finish their development process here, so after phase 1 clinical trials, or here, after phase 2 clinical trials. Why, according to you? That is one of the reasons, but the consequence is that? The consequence is that you have already reached the most important milestone of your project. Going on usually requires more money, significantly, a remarkably more significant amount of money, and are significantly more risky, the phase 3 clinical trials. So the risk-benefit ratio usually prompts pharma startup to sell or to license their assets after phase 2 clinical trials or after phase 1 clinical trials, when they have increased significantly the value of the patent, but the risk of failure is acceptable, okay? Usually this is what happens, it's not rule. Phase 4 clinical trials are something that it's something that usually is done only by big pharma companies, are post-market safety monitoring tests, okay? The aim here, obviously, is to test again the safety and efficacy in a long-term perspective. So what we have learned so far, that the development of a drug is a long process. Usually, again, it's not a rule, it's an average number, but usually this process from the beginning, from the end of phase 3 clinical trials, requires more than 10 years, usually. So it's a long process, and take a look here, it's a risky process. So the risk of failure is very high. So it's expensive and risky. Usually, pharma startups are considered by also by high-risk investors as the most risky startup to invest in. Only specific investors, only, I would say, highly specialized investors in the field of life science invest in pharma startups. It's the most risky business in the life science environment, I would say, in the life science field. And then obviously you have a long time. So if the seed investment is done, for example, at the beginning of preclinical trials, several years are necessary to increase the value of the startup and to have a return of investment. So this is the reason why not all the investors put money inside these kind of projects. One example that that fits very well with this risky concept is the example of the pleconaril. Don't know if you know pleconaril, what is. It's a drug specific for rhinovirus infection that has never entered into market. It was a very promising molecule, a tailor-made molecule that targeted specifically the capsid of rhinovirus. So, it had the perspective of being a very specific, non-toxic and effective molecule. It was tailor-made, it was specific, but in phase 2 clinical trials, it came out that pleconaril selected a remarkably high number of rhinovirus-resistant strains, and that it was toxic, which was something that was not assessed during the phase 1 clinical trials. Why? Because you have a lower number of recruited people during phase 1 clinical trials. So if you have a rare side effect, maybe it's not accessible, it's not visible in that cohort of people. I'm making the example of this drug just to tell you that the path of a development of a drug is full of roadblocks, of possible pitfalls, and some of them cannot be foreseen. So, a risky and an expensive process. Is it clear? If there are any questions, okay. Oh, yes.
How involved are the pharmaceutical companies in the clinical research? Because usually the clinical research is executed by doctors in clinical environment. So how would a drug developer be involved in step 2 and 3?
So usually the bigger pharmaceutical companies are get involved in your startup here or here. Not oftenly here and almost never during preclinical studies. The reason why is that when you reach this point, you as a startup, you have already, how can I say, you have already crossed over the main risks of your business. They usually are involved here or here because they can maximize their investment. Okay. I have never seen examples of pharma startups actively involved in the preclinical studies of a pharma startup. Not here in Italy, anyway. They enter in your development process once you have already fulfilled many milestones, preclinical and at least phase 1 clinical trials. Before, for them, is too risky. They have the money to help you, but they consider the entry in that specific step totally useless and non-valuable for them. So once you have finished the clinical trials, you submit an application to the FDA or EMA, the so-called regulatory review, and then they can give you or not, they allow you or not to put your drug into commerce. So this is the drug development path. We forgot something, actually. We forgot this. Along with the development of your product during preclinical and clinical research, you have to develop a synthesis process that allows you to produce your active principle in a large scale amount. So usually it's a step-by-step process. You start with a lab scale molecule, so a process that can synthesize a few milligrams to 1 kilo of a molecule, and then a pilot scale and a manufacturing scale. Again, also this yellow arrow, this process is associated with specific risks. Maybe you have a very effective molecule, maybe you have a totally safe molecule that, in the case of antivirals, does not select resistant strains. So maybe it's okay here, but maybe you cannot produce your molecule to a large scale amount. Sometimes it's not feasible. Sometimes it's very expensive to produce your molecule. Sometimes you cannot produce your molecule to a high degree of purity. So you have to add the risks associated with these steps to the risks associated with the development of the active principle. But the point is, when I'm at the beginning of my research, maybe I'm here and I have discovered active molecules which are not toxic. I know their mechanism of action, so I have the data to foresee a possible use of my molecules. What do I have to do if I plan to found a startup? How can I understand if my discovery can be exploited to an industrial level? You have to plan your business. So the best thing to do, and this is my second advice, step out from the comfort zone, and the second one is to write at this step during your first steps of the research. When you feel that your discovery is exploitable, you have to write a business plan, which is something you can do, I don't know, in your spare time as a role play game. A business plan is a document which becomes fundamental in the most mature steps of the development of your company. There are many examples of business plans. This is the table of content of the first business plan that we have brought for our startup. So I will tell you something about this. The first chapter of the business plan can be a sort of synopsis of your business idea. So you have to describe your project, including the problem you want to solve, how you want to solve it, and which is your technology. It's not always so easy to describe the problem you want to solve. Maybe you have discovered a very effective molecule, it's not toxic and so on, all the stuff that we have said so far, but you have no problem to solve. Why? Because the pathology that could be targeted, what could be treated by your antiviral, maybe is already treated with other classes of molecules. So you have maybe a good scientific discovery, but you don't have a problem to solve. So you cannot exploit your discovery. The opportunity and the risk. Here, opportunity and risk are terms associated with the market you want to target. Is there a lack inside the market that I want to target? The market is usually generated by the patients of the pathology that you want to target. Are they already treated with other drugs? If not, and if yes, my specific molecule has some specific advantages if compared to the other solutions in the market. Is there an opportunity in the market for my molecule? And which are the risks of my project? Usually the risks of a pharma project we have seen are the ones associated with the development process, which is full of possible pitfalls. Which is the development stage of my idea? So how many of these milestones I already reached? The 4th point is one of the most important ones. I have a team. A team in a pharma startup company means that you have one person that you have people that can manage each step of the development of your company. So inside your team, there must be scientific personnel, but also people with totally different professional skills to manage your IP, to manage your intellectual property, to manage your business. So you must have the team. And I would say this is one of the most important chapters of your business plan when you talk with investors. If you do not have the right people, they do not know who they are financing. They understand that you cannot manage your project. So they won't invest in your project. The market analysis is the 2nd chapter, and this is the most difficult thing for a researcher and sometimes also for a physician to assess at the beginning of the project. So first of all, you must perform a market analysis. So which are those people that generate my total addressable market, and which is the segment of this total addressable market that is targeted, that I can target specifically with my business and with my resources. Which are our customer segments, which is fundamental for a drug development process. For example, at the beginning of our project, we planned to develop a drug for common cold, the first antiviral nasal spray for common cold. We found out during the process that this was not feasible, okay? Because developing an OTC, an over-the-counter drug antiviral drug for common cold requires clinical trials in which you have to gather thousands and thousands of people, which make your development process too expensive, in turns makes your product not acceptable from the point of view of the final price. So we learned that we had to segment our market. So to propose a development process for a drug specific for specific cohorts of patients. For example, asthma patients that must not catch the cold, for cystic fibrosis patients, in general for patients with other respiratory pathologies. So this step to segment your possible customers is an exercise that must be done, I would say, at the very beginning. To identify as sooner as possible your possible market. It's fundamental because it influences both your development path and the design of your clinical trials. One thing is to recruit healthy people that could catch a cold. One thing is to recruit asthma patients that are more easily identifiable. The competitive landscape and positioning, so which is the position in the market that you want to achieve, and which is the competitive landscape, which are your competitors, which does not mean which are those people that sell antivirals from my same pathology. It means which are the other companies that sell solutions, pharmacological solutions for my same pathology. And which is the competitive advantage, which is also known as unique value proposition. So your unique advantage respective to the other solutions. The third chapter is the go-to-market strategy. So from the point in which you are in this moment, which are those steps required to develop your product, which development step you may want to reach, which are not necessarily phase 3 clinical trials, as we said before, which are the resources, financial and human resources you may need to develop your process, which are the channels that you want to use, that you can use to reach your resources. So basically, you have to design a so-called business model canvas, which is this scheme, okay? Which is a one-page scheme that summarizes all these concepts, which are your key partners, your key activities necessary to develop your product, which is your unique value proposition, what makes you different from the others, which are your relationship with customers. At the beginning of a pharma startup process, you do not have relationships with customers. Which are your revenue streams? Again, usually pharma startups usually do not have revenue streams. They only burn cash in the development phase. One strategy that we adopted is to sell services For example, antiviral activity assessment of molecules of other companies, services like this. But usually, a pharma startup do not have revenues. The strategy of the revenue model of a pharma startup is this one: to develop the drug along with step 1, step 2, and step 3, so to clinical trials, and at the same time increase the value of their assets. So the more you develop, the more the value of your IP raises, and then at a certain point, it is economically sustainable to sell your IP or to license your IP or to sell your company that in time has increased its value. When pharma startups are sold to a bigger company, we call this step the exit, which means that the owners of the startup, usually the founders, but not necessarily, has to exit from the startup. The startup is sold to a bigger company. A big exit with a significant return of investment, it's usually the dream of every pharma startup. It's the only way to materialize, to crystallize the value of your company. Is that clear? Which is your sales and marketing strategy, which is something that it's not so easy to understand at the beginning, and which is your product and business roadmap. So basically, this, you have to describe this. Not only. The last chapter are the financials. So how much money do you need to develop your business? As we said before, this process is a very expensive one. I would tell you some numbers, but you have to take it as average numbers, not. So the preclinical research, usually it's the preclinical research needed to fulfill the regulatory requirement, okay, which is always made in good laboratory practices, in GLP, accounts for something like 1 million euros. The clinical research, well, it depends. Phase 1 clinical trials are around a couple of millions of euros. Phase 2 clinical trials around 5 millions of euros. Phase 3 clinical trials, tenth of millions of euros. These are just the costs associated with the preclinical and clinical research. To these numbers, you must add the costs necessary to develop the scalable synthesis process of your molecule, to develop the formulation of your molecule, and then the costs needed, the financial need to manage your startup. So the costs associated with the business management, but also with the IP management. When you have a patent, you have to pay each year fees to maintain your IP. If you don't, you lose your IP, you lose your patent. Again, a risky and expensive process. I don't know if you are confident with these contents. So would you know how to plan, how to write down this kind of document? Because I wasn't able at the beginning, totally. I didn't even know what positioning meant. Positioning is what we have discussed before. So which will be the patients that generate your market. But this is fine. It's normal. At the beginning, you may write down a very naive document, and this is totally okay. The business plan is something that matures next to you. It's a ever-changing document that you want to improve, that you want to edit along with the development of your company, along with the development of your startup, together with the acquirement of new knowledge, of new skills. So everything is fine. Finally, this is the main problem, one of the main problems at the beginning. You have a scientific and regulatory process that must be fulfilled in each one of its milestones. But if you have a company and you have a patent, you have to manage also your IP, you have to manage your business, and you must know how to develop. So which are the activities needed to develop your business, to identify the segment of market that you want to target. So how can I do something like this? You cannot do everything by yourself, obviously. You need a team of people, each one with its specific skills. Not necessarily a tree and a raccoon, but people that are able to do their job, which means people that can manage the development of your synthetic scalable process, that can manage the development of a formulation of your molecule, to manage the pharmacokinetics and toxicology studies. All these people, this is fundamental, all these people should not be people from academy, and usually are not. Usually the people that are able to manage these specific activities are people that have matured a specific experience in a company. So people that come from the industry. This is usually what happens. People that must manage business issues associated with the IP management, for example. So you need lawyers. You need a business developer, which is the person that identifies specifically your segment of market and knows how to reach that specific market. A person that has a very specific a network of, for example, potential investors or industrial partners. In this process, in general, for all the companies, a network is fundamental, and engaging people with a network it's fundamental to have. You must have a market analyst. But most important, this is one of the most important lessons I learned. You must have a pharmaceutical regulatory affairs consultant. You may make the error to think that you will need a person like this maybe in the later stages of your development process. This is an error. A regulatory pharmaceutical consultant is needed at the beginning because a person like this, a professional like this, will indicate to you very specifically which are the regulatory fulfillments necessary to develop your product. And he will tell you that it's feasible to develop your product for that specific cohort of patients. It will tell you, well, maybe the clinical trials cannot be designed for your specific therapeutic indication. So you must associate with a person like this at the very beginning of your process. Don't make this error. This is a very frequent mistake. The problem is that you have to pay these people. And you have to pay for your IP. You have to pay the fees generated from your company. You have to pay for the development, so for the clinical trials. So talking about financial needs, which are the main funding bodies that are involved in the pharma startup development? Well, at the beginning, you may rely on grant fundings. When you are still in the stage of your research, for example, so in the discovery stage, you can rely on grant fundings, which are important because usually you submit a research proposal, and if the funding body, just like the NIH, Ministero dell'Università e della Ricerca in Italy, and the ERC, if they accept your proposal, they provide you with financial resources with nothing in exchange. So they just give you the money and monitor the milestones of your project are fulfilled. Okay? It's fundamental which means that you do not have to dilute the shares of your company. Or it means that you can fund your research if you don't have your company. Maybe you have a good idea, but you do not have yet effective proof of principle of your molecule. This is the stage to apply for the grant fundings. But at the certain point of the development of your startup, which is usually during the preclinical phase, you have significantly more relevant financial needs. So you have to submit your business plan to private investors, just like, for example, business angels and venture capitals. Do you know, are you confident with these kind of terms? Okay. Business angels are not angels, are more like sharks sometimes. Business angels are individuals with a portfolio of investment, with a significant portfolio of investment, so they can risk an amount of money on your project. Usually, the amount of money that they risk on your project is around 100,000 euros or around this number. Okay. By contrast, venture capitals are associated funds. So many people that put money in a fund that is managed usually by another company, another entity. So they have more relevant financial resources. Okay, usually they can fund up to 1 million euros of money to your project. So usually this is what happens. You need the grant fundings at the beginning of your project. Business angels come during the preclinical research, and venture capitals associate with your company only in later steps. This is usually what happens. It's not the rule, okay? Sometimes, even at a more mature step of your process, you may want to apply grant fundings, okay? The idea in this case is just to integrate the financial resources that are provided to you from business angels and venture capitals. But I did not tell you an important thing. What do they ask in exchange? shares of your company. The point is this: the more your company is risky, the more your company is in the earliest stage is your company, the more is the shares that they ask to you. At the very beginning of our path, we were in the discovery phase, but we already had a company. We ran into a venture capital fund, and they asked to us the 67% of our company to fund our project. I won't tell you this is a rule, but it's usually that something happens. So venture capitals, usually when you are an early stage pharma startup, they want to acquire, if they want to fund your startup, they ask you for the majority of equities, the absolute majority of equities, the 67%. So the later they, and from your point of view, from the point of view of the early owners, of the founders of your company, the later they associate to you, the better it is. When a business angel or a venture capital enter inside the shares of your company, the term that it's used is that they dilute your shares. So the value of your shares decreases significantly because they own the majority of shares. Another thing they usually require, more frequently venture capitals, is a board member position. So they want to decide. So they usually take over your process, which is something good, okay, sometimes, because they provide you both with monies, but also with expertise, okay? So it's not so bad. I won't provide you with this message. It's not always so bad. But the point is, which are the correct business angels and the correct venture capitals to talk with if you have a pharma startup? There are, because the point is that there are many business angels and venture capitals specialized in life science projects, but only a few of these funding bodies are specialized in pharma startup. Only few of them, because the development of a drug require highly specific skills. As I told you, from the regulatory point of view, from the industrial point of view, from the point of view of the knowledge of the market. But most of all, a pharma startup requires a high amount of money. The return of investment is shifted from the seeding point of your startup of several years, and it's a highly risky business. So only a few amount of life science specialized business angels and venture capitals fund specific pharma startups. And it's a very competitive field. So the problem is, how do I convince these people? The main problem you have when you talk with investors, usually, I don't know, but this was my problem specifically, is that I didn't even understand what they were asking to me. Let's see some questions. Okay, describe your IP status. It's difficult, but sometimes you have the skills to answer. I patented, I did not patent. My invention is patented in these countries, Europe and so on. So, this is not the most difficult question. Which is the problem that is addressed? Easy. Which are the existing products and technology that will be replaced by your invention? How big is the potential market? Here they want to know the numbers, not the number of the people that generate your market. They want to know the number of euros or dollars that are generated by your market. And it's not an answer that you have in your pocket. Are there any other competitors? And it's always difficult to, the world is big, okay? And so it's not so easy to identify which are your main competitors, which are the other people that sell a solution for your same problem, which are the risk and probability of success. They want a number. They want, more oftenly, they want a risk assessment analysis, which is not something that a researcher, I speak from my point of view, personally, which is not something that a researcher usually can do. Is it possible to scale up your productive process? So can you produce your molecule to a large scale? Even Fleming did not receive a Nobel before a scalable process for the penicillin was invented. Maybe you have a wonderful scientific discovery, but it's not exploitable to a commercial level. Which are the regulatory issues? So, which is the sign and how you want to design your clinical trials? Which type of people, which segments, which cohorts of patients you want to recruit? Which means usually you want to develop an OTC, an over-the-counter drug, or a prescribed drug. It's not so easy to assess at the beginning. Which is your budget? How many equities you want to sell to me, to the investor? And which is the pre-money evaluation of the company? And this was one of the most difficult questions. They asked to us, the first chances to meet investors, they asked to us, which is the value of your company? I don't know. Pre-money evaluation of your company is the total value of your assets before you received the first seed investment. How to calculate this number? There are several tools to make this, but these are tools that can be managed to specifically skilled people. Okay? It's not something that can be done by a researcher at the beginning of his scientific process. These are usually calculations, the pre-money evaluation. It's usually a calculation that it's made on the basis of the market, on the dimensions of the market. Which is the cost of licensing, of renting your patent, or the selling value of your patent. So these are usually the questions that an investor asks you.
You mentioned over-the-counter medication and prescribed medication. At which point of at which step of the research is this decided, and basically what are the pros and cons on putting your medication on either?
As sooner as possible, because the aim to develop an over-the-counter drug or a prescribed drug influences all the development process. One thing is to, I come back to the example of my startup. One thing is to develop a drug for the common cold. So an over-the-counter for healthy people. Totally different topic is to develop a drug which is prescribed only for asthma patients. In the first example, in the clinical trials, you have to recruit healthy people, hoping that they catch the cold. which is not so ethically good, but another thing is to develop a prescribed drug. In the clinical trials, you have to recruit patients with asthma, for example, or with the COPD, or with cystic fibrosis. So the sooner you have this information, the better it is. This is why you have, even in the discovery phase, you must reach a regulatory affair expert, because you want to, you may want to plan a specific development roadmap of your product. So even at the beginning, you must know how you want to design the preclinical stages, but more importantly, and it's more difficult, the clinical step of your roadmap. And in order to design these steps, you must know which type of drug you want to develop at the very beginning. Which is an error, again. I'm sorry, I will repeat, but it's very important. It's usually the error that we do as researcher, okay? The exercise you may want to do when you write down your business plan and you imagine your business, the exercise is to start from the end of the process and then plan all the other steps backwards. This is the exercise, which is exactly the opposite that we do with our research. Start from the end, so from the market, and then go backwards at the beginning.
You are a researcher working for the University of Turin, but does the university own part of something that you will discover?
Very good question. In my specific case, no. I own my patent together with other people. But this can be done only in Italy, where there is a so-called professor privilege. You work for the university, but if you want, you can patent your discovery if it's patentable and pay the cost by yourself. So you have honors, but also fees to pay. This is a specific legal issue in Italy. In all the other countries, this is not feasible. If you work for a university, the university owns your patent. You can be the inventor, okay? But you do not have economical rights on your invention. Is this clear? There are two types of figures associated with an invention: the owner and the inventor. The owner is the one that has the property of the patent, which means that it's also the one that will have the economical benefits, but also has the duty to pay the fees, the annual fees of the patent. The inventor is the person that generated the father of the patent, but it's the intellectual father. So has no economical rights on the patent. So it's very different. In Italy, if you work for university, you can be both inventor and owner. But in all the other countries, if you work for the university, you can only be the inventor. Yeah.
Does this special situation in Italy make it like the equivalent of tax havens in the business tax havens? It's like countries that don't have any taxes and people send their money there. It's similar to a scheme in the business world where people want to. Since this special thing in Italy where you can be the inventor and the owner of whatever you invented, do people from all over the world come to Italy to use this, like as a escape plan or something?
Absolutely. No, absolutely. You have to patent your invention in your country, and then during the end of the first year of your invention, you have to decide which other countries, in which other countries you have to submit your patent application. So at the beginning, you always have to patent in your country. Usually, it's not even feasible. I mean, if I was a US professor, I cannot even submit a patent application. It's my university that submits my patent application. So you don't even have the problem to decide in which country do I have to patent. It's your university that owns your patent. So you cannot choose anything.
At which point of the development process do physicians come into play?
It depends. Sometimes they are involved at the very beginning during the inventive process. You know, maybe sometimes the discovery process, so the step one, involves clinical trials, maybe funded by grant fundings. So in that case, it's not hard to imagine the involvement of a physician. Actually, in our startup, we are 3 owners, and one of them is a physician and is with us since the beginning of our journey, because he's an expert of oxysterols, basically. So a physician can enter into this process even at the very beginning. It depends. Yeah.
My question is related to previous slides. Even grant fundings ask for, such as MUR or EMA, ask for share of company, because as far as I understood, it was supposed to be non-profit help.
Usually they are non-profit, so usually they don't ask for shares. But there are specific grant fundings from the EU Commission that specifically fund companies. And I guess that in that case, they ask a share of your company. That's specific. But in general, all the fundings that fund research projects do not ask for shares of anything, because sometimes they do not even fund companies, they fund researchers. So they just monitor your project. So in order to receive the money, you must fulfill the milestone of your project. That is required, always. That's the point. So, many questions, and you don't know how to answer, which is the same problem when you have to write down your business plan. Several contents, and you have to fill all your paragraph, which is fine. It's fine at the beginning. So my advice is to, at the very beginning, to challenge your idea. To challenge your business plan, okay? So to take part to startup competitions. It's a good way to challenge your idea, and it prompts you to write down a business plan. Competitions challenge you also in terms of communication, okay? So how to communicate your idea, which are the contents of your idea that you may want to deliver to convince an investor. So startup competitions, some of them provide you with a mentor or an acceleration program. which is a step-by-step program in which different professionals with all the skills that you have seen in the last slide are involved. They can give you advice. Sometimes when you win the competition, you can win consultancies that you can receive from these people, which are quite useful sometimes. Sometimes you can also win money. So it's also a way to fund your idea. My advice is to take part to these kind of events. I have actually two advices. To take part to these kind of events at the beginning, when you have to challenge your business plan, basically, and your idea. Second, choose carefully. One of the associate of the company that I own told me oftenly that most of these startup competition are circuses, okay, are made only for the professionals involved, not for you. So I give you three advices, at least taking into account the ones that I have experienced here in Italy. BioHapper, the last event was, the last call was in 2020. 2020. BioHapper is a startup competition with an accelerator program which is very effective, which is managed by the Politecnico of Milan and Novartis. It's a very effective accelerator program. This is the competition that, and this is the program that has taught us the most part of the things that we have learned at the beginning. How to write down a business plan, how to communicate our idea, how to identify the market, and so on. The Startup is a series of competition which are held in any region of Italy each year. So there's a call each year. It's a business plan competition, just like the PNI, the National Innovation Prize. Both of them are, so BioHapper helps you to write down a business plan fundamentally, while Startup and PNI are for those companies that already have a business plan. It's a business plan competition. Very useful. The other thing to do at the beginning is to prepare a pitch deck. So a brief presentation of your business, of your startup. Must include specific contents that I'm not able to let you see. Anyway, must contain specific contents. For example, the problem you want to solve, which is your solution, which is your market, which are your competitors, which is your unique value proposition, and you have to deliver these contents in 3 to 5 minutes. Okay? It's not a scientific communication, it's totally different. Here you do not have to communicate data to an audience, to a critical audience. You want to sell something, okay? So just to give you an idea, and this is the last thing that I leave you, just to give you an idea of what a pitch presentation is, I let you see the pitch of my company that I delivered in, I guess, 2017 or something like that. Meanwhile, if there are questions, just ask. Oh, yes. Another thing is that usually universities have incubators. have startup incubators. Most of the times at the beginning, it's a good idea to talk with them. Yeah, sure.
So, apart from Italy, what is the incentive of a researcher to be an inventor? The incentive. There is no financial incentive. If the university owns all the patent, you will just be a figurehead.
That's another good point. I would say that abroad there is no economical reconnaissance, but
Sintomi del raffreddore e dell'influenza? Tachiflu, Task.
you can be. This is the commercial, not of the company, obviously. You don't have the economical revenues of your project, but you can be part of it. You can manage the development of your startup. And you are a scientist, so you can, by means of having a patent, by means of having a company, you can be yourself part of the development process of your discovery. So in this case, you don't have, obviously, economical revenues, but you can do something very concrete. Okay? Don't know if I answered to your question. Yes, I know. You don't own anything, but...
I mean, down the line, when you will need to get the angel investors or the venture capitalists, will you make the decision, or will the university make the decision? Okay, so that's the...
Yeah, it's usually the owner that has, how can I say, that must gather the investors, recruit the investors. It's always the owner. Mm-hmm. Maybe you are still inside your startup with a position as a scientific manager of the activity, yes. But the ones that manage your IP portfolio in your business is the owner of the startup, the owner of the patent. But it's like this also in Italy. At a certain point of the story of your startup, you must delegate this activity to somebody else. You cannot do anything, basically because you cannot do it. You don't have the skills. And you have to recruit these people early, very early in your project. Okay.
So, good afternoon. I'm Andrea Civra, co-founder of Panoxivir. We will develop the first cure for the most widespread viral disease, the common cold. You may think that a cold is not a big problem. You have to think that we experience it 2 to 4 times per year, in children even more. So if you take into account a mean duration of symptoms that goes from 7 to 10 days, we have to conclude that we spend something like 5 years of our lives together with common cold. And the social cost is not cheap. $40 billion per year only in the US. Why? Because we don't have a cure for common cold. It's a huge medical need. The available common cold remedies only ease some symptoms, but they do not shorten the duration of the pathology because they do not eliminate the cause of the pathology, which is a virus, the rhinovirus. So we should have an antiviral. We don't have, because there are more than 150 different serotypes of the virus, and an antiviral should be active against all of them. We have a solution. It's called Panoxivir, and it will be the first antiviral nasal spray to treat and prevent common cold. The active ingredient of Panoxivir are the oxysterols, molecules that we spontaneously produce in our body to fight viral infections. They do not act on the highly variable viruses, they act on the site of the infection, the nasal mucosa, by getting it resistant to viral infection. So they have a wide spectrum antiviral activity against all the rhinovirus serotypes. If compared to the available common cold remedies, panoxivir will be able to cure the pathology, but also to prevent it. Prevent the symptom, obviously, but also preventing the spreading of the pathology. We're talking about an infectious disease with a positive impact on the healthcare costs, and with an impact also on the common cold remedies market, which is accessible overall in the US and in Europe in 10 billion euros. This is the development roadmap of panoxivir. Won't go through details. Usually, pharma startups do the best exit after phase 2 clinical trials by selling the IPO or selling the company. We have patented the use of oxysterols against human rhinovirus, papillomavirus, and rotavirus, 3 important pathogens. By now, we have developed the industrial and scalable manufacturing method to produce our molecules along with their formulation, and we have validated their antiviral activity on human nasal mucosa, so we know they are non-toxic and they work, they really work. An ambitious project, but we have won several awards in national and international competition, and we have assembled an interdisciplinary and high-skilled team to develop all the activities of panoxivir. Together, we will give you the chance to set you free from common cold. Thank you for your attention.
So, science content, 0. We were trying to sell our idea. So in, I don't know how much, 3 minutes, we had to deliver all those contents that we have talked about so far. The spoiler is that I didn't win that competition, but anyway, it was good to see at the end of the presentation. So thank to all of you for being here, and I hope it's been useful. You're welcome. If you have any other questions, even after, just write me and I will answer, okay?
Okay, so a quick word, everyone. Guys, before you leave, if you can just scan this feedback form and fill it, it will take like a minute or 2. It will really help us for future events. And thanks again for being here. It has been a pleasure. Thanks a lot, Professor Chivra.